Good morning, Your Excellency, ladies and gentlemen. I'm Richard Selman and I work for DEFA. I'm very delighted to welcome Dr. Arno Wuenschmann, all the way from Minnesota this morning to speak to you. There's clearly been a long-standing interest in wallaby issues on the Isle of Man, with feral wallabies out since the early days after the Curragh Wildlife Park opened in the early 1960s. And I can mention a little bit of the background to set the scene before Dr. Wuenschmann speaks to you. Our first discussions on wallaby issues resulted in a master's student that I invited to the department back in 2008, Andrew Harvey from the University College in London. And he made a census based on transects in the area that they were then known from back in 2008 in Curragh. This showed about 100 animals at that time, and he also looked a little into the diet and the effects of the population on the ecology, which were not considered to be greatly damaging at that time. Then in 2017, Paige Havlin, an honours student from Queen's College Belfast, worked through Manx Wildlife Trust using a different method with cameras and came up with a similar figure for Curragh. But then she extrapolated this figure across the island to the extent of an area where public sightings had been made around about that time and produced a population figure which was published of 1,742 animals plus or minus 455, so quite a bit of error in that. Obviously, a considerable difference there. Some of us thought that was rather unjustifiable extrapolation of the data that was then available, but we did know that wallabies wandered from end to end on the island, seen Port St Mary, Point of Ayr. They certainly move about. The Manx Wildlife Trust then undertook a thermal drone photography census of Curragh with some further work on diet and gut fauna. And they found significantly more animals than previously, with 560 just in and around the Curraghs. So this got us thinking, well, what's going on? We felt that we needed more information at that point on both the overall population in the island and on its health status. The department therefore contracted the Wildlife Trust in 2024 to undertake an island-wide census of the wallabies and the goats. And that was subcontracted by the Wildlife Trust as the previous work to BH Wildlife Consultancy. The report is published online, so you can go and have a read there if you're interested. And a figure that came out of that of almost 1,000 does seem reasonable and relatively reliable. There were 869 actually counted individual animals. So this, surprisingly, is actually not so hugely far from Page's figure, if you take the large errors into account. So we now know that there's a large feral population on the Isle of Man, but its health status remains an enigma to us and the topic of much public debate. In considering the effects of this growing population, the department is interested in the issues for human interests, such as public health, agriculture, nature conservation, for landowners, and for animal collections. And there's a significant gap in our knowledge on the health status of feral wallabies and how this may affect those interests. The work here to be presented by Dr. Wuenschmann provides an early look into that world from an animal health perspective. And so Dr. Wuenschmann graduated as a vet from the Justus Liebig University in Gießen in Germany, where he went on to graduate as a doctor in veterinary medicine. He's board certified in veterinary pathology by the American College of Veterinary Pathologists, and since 2000 he's been a diagnostic pathologist at the Veterinary Diagnostic Laboratory in the Department of Veterinary Population Medicine at the University of Minnesota in the United States. And he specializes in wildlife and exotic animal pathology. Although he is a diagnostic pathologist with experience in a broad range of domestic species and their diseases, his primary focus is on infectious and toxic diseases of wildlife with numerous publications in the field. He has worked extensively on birds of prey, being affiliated with the Raptor Center of the University of Minnesota, but is equally interested in birds, mammals, reptiles, and amphibians, whatever else life throws at him. His wide-ranging work has included infections in deer and moose, metal levels in bald eagles, and garter snakes. Herr Dr. Wuenschmann, Sie haben das Wort.
Thank you very much. Well, again, thank you very much for this warm welcome. Beautiful to see so many of you here having an interest in this topic that is also, again, close to my heart, being a wildlife pathologist. I don't know, if you have any phones, I don't know, maybe you want to silence them. Just wanted to maybe throw this out there. The other question that I have, can you hear me in the back? Because if not, I will use the phone, but I am kind of a wanderer a little bit. So, okay, well, thank you very much again. And so with that, maybe let's just jump right into it. So we're talking about the health status of wildlife populations today in general, how one could examine those, and then more specifically about the wallaby population here on the Isle of Man. And before we get to that, maybe some of you are wondering why I am here from Minnesota, and so I will try to explain it just a little bit. There's also some of the pictures that I'm going to show, that is the nature of the beast, so to speak, are going to be maybe upsetting to some people, not to me, but upsetting to some people. So just a slight warning here. I guess I work, as was mentioned by Richard, thank you very much, at the Veterinary Diagnostic Laboratory at the College of Veterinary Medicine in Minnesota. And yes, I am a pathologist since about 1993 and board certified since 1999. And my special interest is diseases of wild animals, exotic animals, and zoo animals. We live in Minnesota. I'm not sure how many of you are familiar with the map of the United States. We are bordering Canada, so we're all the way up there. And it's also the moniker for our state is the land of 10,000 lakes. So basically everybody who lives there also gets a lake. That's a nice benefit. And we do in Minnesota, that's one of the reasons why I like it there so much, cherish our wildlife. And so there is one, is the moose, loons, you can see that. There's a lot of fisheries in, freshwater fishery in Minnesota. We are also at the Great Lake, at Lake Superior. And well, this is the archetypical Minnesotan as people imagine them to be where we are. Has these pleated shirt and an axe in their hand. So that's Paul Bunyan. And yeah, this is where we live. So we do have quite a bit of nature around ourselves. And we get to see a fair amount of wildlife. And so this is my lovely wife who is sitting in the audience and our beautiful little dog. This is our cat that is on the prowl here right now. Maybe he shouldn't be out there. He should be showing us probably here at the Manx Wildlife Trust event. Then we have some cats, horses, and we have another dog with us. So this is where we live, and this is the family. And again, I mentioned we do see wildlife quite a bit where we are there, all sorts of monarch butterflies, fascinating creatures. They actually over I believe 3 generations migrate to Mexico and back. Yeah, we have newts or salamanders, fish obviously, a beaver dam living down on the lake where the coyotes pass. This is not a wolf, this is a coyote. We have otters, bald eagles. You name it, there's quite a bit. So I live in wildlife paradise almost. But we do also love the Isle of Man, and so that's maybe why I'm here. My wife and I come here on a regular basis. We love, well, pretty much everything about this island. And so we love the glens and the old historic sites, so prehistoric sites, coastline, all of this, and for some reason we are drawn to Peel. So we spend time in Peel on a regular basis, and it is very fortuitous that the Manx Wildlife Trust is sitting in Peel, and so yes, there was a connection made. So now I want to talk to you briefly about values and pitfalls of wildlife disease monitoring, and I want to use a study that we have conducted in Minnesota on moose as the segue into the topic. And so, Wildlife obviously lives here at the intersection of animal health, human health, environment health, and ecosystem health. And so why would one even consider doing wildlife monitoring? Well, I guess one reason would be for the greater good. I mean, I think we share the planet with our wildlife, and I consider it a moral duty of ours to take care of wildlife. But that is not an argument that sits well with everybody, as I have come to understand over the years working on wildlife. So there are also other reasons why we would want to look at what our wildlife, what the health status of our wildlife is. And some of it has to do with public health and food safety. I mean, wildlife obviously carries diseases that also can affect our livestock, our pets, and us. And so there are many examples of diseases that I could mention here. I mean, we just have to think about highly pathogenic avian influenza virus. I mean, there is, it has spread to humans, but it's not transmitted from human to human yet. But we are very worried about the next mutation that may allow that virus to spread from human to human. That could be absolutely detrimental. And of course, this highly pathogenic avian influenza is maintained in the bird population, and as we know now, not only in the bird population but also in mammals. And we only know this because we look at these mammals and we dissect these animals and conduct testing of these animals. And so now we know, yes, the mink may have it, foxes may have it, and all sorts of birds get it, not only poultry. Raptors died from it in large numbers and are still dying from it. Other things is chronic wasting disease, for example, in North America. That is a disease very closely related to mad cow disease, so a spongiform encephalopathy. And so we have to keep an eye on the deer population to see where this disease is. All of these are reasons why we should really be interested, even if it was not only for the greater good, but actually from a human and animal health perspective. Then there are legal concerns when wildlife pathologists or wildlife veterinarians and biologists become involved. We had a few tragic events. We had suddenly a few wolves and bald eagles dying near a landfill in Minnesota, and it was a little bit unclear why that happened. So they were submitted for a post-mortem examination, and as it turns out, on this landfill there were some chemically euthanized animals that were disposed of improperly. And so those eagles and wolves had access, and probably other scavengers had access to these carcasses, and they died. While most people are probably not so concerned if a crow dies or something, if a bald eagle or wolf dies in the United States, well, that is a federal crime because those are federally protected animals. And so we looked into this. But again, there are other examples. Poaching, of course, occasionally we get animals that were poached. And in order to strengthen the legal argument, a post-mortem examination is going to be very helpful. Another interesting case we had were some Canada geese and swans that were found dead on a lake in Minnesota on the ice. We do get ice up there quite a bit. It's actually quite a cold state, but that in some ways can help because it preserves the animal better for the examination. But they were there in fairly large number, and at that point the high-path avian influenza outbreak had already begun, and everybody thought, okay, this is high-path avian influenza virus. As it turned out, when we looked at these animals, no, it wasn't high-path avian influenza virus, but it was actually a fungal disease of the lungs that caused the demise of these animals. I mean, I received 4 of these animals, and all 4 had the same problem. And so what had happened is that there was a train track next to the lake, and they had spilled some corn. And on the corn there was a mold that was growing, and this mold, aspergillus, called aspergillus, actually was taken up by the geese and rapidly killed at least 40 or 50 of them and a few swans as well. So there are reasons why you would want to look at wildlife from a legal aspect. And of course, I have to admit, I'm also a veterinary pathologist in academia, so we also look for publishing opportunities, and there's a lot of new things that can be found in a wildlife population, particularly the ones that haven't been looked at before. So, who participates in a wildlife health and disease monitoring program? Well, it's obvious that there are the wildlife biologists involved. Obviously, there are veterinarians involved. Depending on what kind of study you conduct, this can be clinicians and can include veterinary pathologists. And I guess I may be biased, but veterinary pathologists might provide the biggest bang for the buck, I would think. But we might talk about that later. But a huge part of this, of course, is the public. I mean, the public has to be involved. The public has to be involved in what kind of questions are being asked and why are we doing it from one perspective. And I mean, I will also probably say from a funding perspective, but then also on a very practical level. Well, the public gets a lot of eyes on the ground, on the landscape. They know a lot. So then wildlife biologists and certainly veterinarians cannot find these animals out in the open, but the public knows where they are and can report them. So that's also a very important aspect. So there's a little schematic I did. Well, I circled post-mortem examination, so the part where pathologists become involved thick, again, because I think it's very important and a very good starting point. But there's also live animal work that can be done, and maybe we'll talk about that in just a little bit. But of course, as a pathologist, we look at what kind of diseases we find in the particular animal that is in front of us. And then, based on what we find, we can submit samples for microbiology, so bacteriological testing, virology testing, parasitology testing, and potentially some of the newer methods, the molecular methods that can be very helpful. We can look at the gastric contents and can kind of try to figure out, and that would be a biologist probably that would be doing this. We collect gastric contents, and then once somebody can decide, well, what are they actually feeding on, and what are they, what can potentially cause also toxicity in these animals. So that is another question, and we can examine tissues for toxic compounds. In the case of the wallaby population, obviously it would be very interesting to get a geneticist involved, because you would assume that over time, or that there is significant inbreeding in the population that might cause malformations or some other diseases in these animals. And so in order to determine that, of course, you have to look at enough animals to get a sense of what's going on. You can look at the reproductive status of animals, because, of course, a population lives from the recruitment of young into the population. And so you can draw some conclusions by doing both post-mortem work, but also particularly live animal work on what the reproductive success is in this population. When we think about live animals, well, there are now very advanced techniques where you can follow live animals. When you collar or tag them, you can put GPS collars on them, and then you know exactly where they're going. And by doing that, you can also a little bit avoid the bias that you get by looking at animals that are just randomly found. So, for example, if you had a neurologic condition in a certain population, it's far more likely that you're going to find those neurological animals that behave oddly, and you can, the public or whoever can notice this, right? So there is if you just look at the dead animals that are there or that have been reported, there is a risk of a bias of your population. If you collar animals at some point and you investigate every animal that was collared, well, that is not as biased as a population as the one that you just find or is being noticed by the public. And we heard this just in the introduction, with any wildlife population, the question is, well, how many are there, right? And so that is the first thing that you can do, is try to estimate how big the population is, and there are various methods. And in our case in Minnesota, over time they have been optimized. The counting efforts as well as the modeling has been optimized. So we have a fairly good idea, and the Isle of Man now has a very good idea of how many animals are actually out there. And then if you had a live animal on your hands, you could actually draw blood and look for antibodies against diseases, and that would tell you which disease agents the animals were actually exposed to. It doesn't tell you whether they died from that condition. That's something, again, you have to figure out at a post-mortem examination, but it tells you, well, what are they actually susceptible to, and how often have they been exposed to these certain agents. So all of these are things that we can do. I will say the live animal handling is, of course, bears a certain risk. So if you capture animals, there's a risk that some of them get injured, just trauma, right? Or that actually develop a muscle disease after they have been caught and die within 1 or 2 weeks of being caught. And so in our moose study that we conducted, well, it was basically the loss of 2 to 3% of the animals was accepted. Not by everybody, as it turns out, but by the scientists. Okay, and then I just really briefly want to circle back to pathology. It's basically the study of diseases. And when you are a pathologist, you conduct a gross examination of the animals with your naked eye, so to speak. But most of what pathologists do is they hover over a microscope. Organ samples are being collected from the animals and then cut so you can actually look at these organs under the microscope. And that gives you a lot more information than just your gross examination. And again, based on what is being found, we then can submit samples for testing if we actually thought of collecting those samples, of course, right? So there are some pitfalls to pathology, and so some of them are, well, the carcasses or the bodies that we look at, they should be well preserved, because if they're not well preserved, there is not an awful lot a pathologist can do with it. And so time is a little bit of the essence. In this kind of animal that I saw in closed sard field, well, I mean, the skin is coming off, it's bloated, and this animal is no longer going to be suitable for a histological examination. Sure, you could still figure out whether somebody shot it or whether it had a skull fracture or something like this. It's going to be unpleasant, but it's possible. But you cannot do any further disease testing really with this kind of carcass. And then my wife actually played around with ChatGPT. I don't know whether you're familiar with this one. And this is what comes up when you punch in wallaby and pathologist and scavengers, because that's basically what a pathologist is. We compete with the scavengers for our fresh tissue sections. And it almost looks a little bit like me, I think. Anna said it shouldn't look as angry as it is, but I said, no, that's okay. If you stand up to a spotted hyena, maybe you need a little bit of rigor here. Okay, so the other problem are scavengers, particularly where we are. This was a deer that was seen the day before, completely intact, freshly dead. Unfortunately, it took us about 24 hours to get to it, and this is how it looked after 24 hours when the coyotes got into it. And, well, there are no internal organs left that I could actually collect. And And then there are costs. I will not deny it. There are costs involved with pathology as well. There's a laboratory. Usually you could do field necropsies, we'll see that. And then the pathologist costs money. And so, of course, there needs to be a certain funding if you want to be really serious about it. So when do we employ wildlife pathology? Well, there should be a specific question or specific questions that we're going to address. And obviously the stakeholders, public and the pathologists should agree on that question. And then we can move forward if the funding is there. Again, in any time of legal concern, it's important for wildlife pathology work to be involved. And so, as I mentioned before, we need freshly dead carcasses to do it. And again, I mentioned I want to use the example of the moose. It's an iconic species in Minnesota, an umbrella species, so to speak. So many people are very interested in it. And if you protect the moose, you protect a fair amount of the habitat as well. But in Minnesota, we noticed, or some people noticed in around the late 90s, early 2000s, that the population was decreasing. So it's obviously a different starting point than here, the wallaby population. So we had two populations, one in the west of the state, northwest, and one in the northeast. The one in the northwest actually basically is non-existent since early 2000 anymore. And these are the figures for the population in the east. Again, there was a different type of counting employed, and later on they decided that the helicopter counting, very expensive of course, is going to be better. And so these animals are counted in the winter when the foliage is down and you can actually see them. There's a spotter plane on top, and then there's a helicopter plane that also counts these animals. Actually, what I'm mentioning here right now is for the collaring efforts. There's a spotter plane and then a helicopter. But you can maybe appreciate the fairly dramatic decrease of the population. It was basically halved over about 8-9 years, and that had some Minnesotans up in arms. And they wanted to know why this is happening. And so there were several hypotheses that were thrown out there. This may have to do with heat stress. Heat stress would potentially increase the mortality rate of moose in Minnesota, and it would potentially also decrease the reproductive rate of moose in Minnesota. Then there was a thought that maybe the increasing wolf population were a factor in this decrease of the population. But then there were other people that thought, well, maybe they have some diseases that are popping up in them and they are new and they are impacting the moose. And so that's where basically I came in. So this is a term that the biologist had coined in the late 90s. They called this tip-over disease. They found a moose that was otherwise looking perfectly fine. There was no evidence of a struggle, a wolf kill, predation, or anything like this, or a bear kill. Although the bear, they focus more on the moose calves, and the wolves, they focus on calves and on the adults. So there was no struggle of that, and so they called this tip-over disease. Animal just was there and tipped over and was dead. Then they did a study, the biologist did a study, and a very well executed study. And so they noticed that the livers look clearly abnormal and that there is a parasite in the liver fluke, the North American liver fluke to be precise. And the final host for that North American liver fluke is the white-tailed deer. And so occasionally other species become infected, cattle, sheep, llamas, alpacas, but also moose. And moose become infected quite commonly, and so there wasn't really a lot of historical record about this. And so this is a liver that is utterly abnormal, just like this one, as you can see when you compare it to a normal looking liver. So the conclusion was that, well, the liver fluke is the culprit, or I mean at least is a culprit. And that would lead us somewhat to the white-tailed deer and the snails, the intermediate hosts that have to be there. And so one would have to look at the white-tailed deer population and those intermediate hosts to see what the burden is. And so I looked at the white-tailed deer. There's actually surprisingly little data on the white-tailed deer population, but anecdotally it had increased over the years in Minnesota, even in the really cold boreal areas here of northern Minnesota. And this is just based, in this case, on deer hunting success. So it's a very rough estimate. It's not like somebody went there and counted them. But I mean, we noticed that there's a large number of deer hunted in these northern counties, over 1,000 in certain sectors of these counties, and so we know there are a lot of white-tailed deer around. Okay, so there was one outcome of this early study. Now, then a little bit later, the Department of Natural Resources with Dr. Carstensen decided to do a more thorough study of the population. And so they set out to optimize the counting effort and models, as I mentioned, and they wanted to collar or tag moose with these GPS modern collars that would tell them where the moose are at certain times. They actually recorded the movement every 6 hours. You could do this every hour if you wanted to, but then the battery life of the collar is a limiting factor. They wanted to monitor these moose at least over 5 years. Unfortunately, as it turns out, the first collars they used, some of them were non-functional really, so they spent a lot of money putting collars on moose, and then because they didn't function properly, lost a fair amount of data. But all of this got better over time. And so the movement data are really interesting. They didn't have those before 12 years ago. But you can basically, if you get accustomed with them, you can figure out, well, where do these animals spend the most time, right? Where do they forage? Things they didn't really exactly know. And also, well, where do they give birth? The moose in Minnesota give birth around mid-May. And so you could see where these cows sequester during that time of the year and that time period, and then they could pinpoint, well, this is the habitat where actually that is suitable for giving birth, for example, right? And so they could also do a reproductive assessment. They could go to these sites where the moose were, the cows were at the time, and could see how many calves were being born. So all of this very interesting data. And then the collars have a death signal function. Anytime a moose doesn't move for 6 hours, you have to assume it's dead. And so there were some funny observations where it was not moving for 6, 8 hours, and then it was moving again. And they thought, this moose is probably not dead. Somehow it must be an outlier. As it turned out, these were moose that were killed by wolves, and the wolves suddenly moved around the collar, and so that gave it a signal, and so we lost that moose to follow up. But so we knew where the moose were dying, and they knew where the moose were dying, and so we could actually either send a team in of field biologists, usually into the field, and collect samples and do the examination, and or they could recover the carcass entirely, which is a fair amount of work in a moose case, and take it down to our lab, which is about a 4 or 5 hour drive usually. I mean, the moose, we know a fair amount about moose in the meantime. You can actually analyze the teeth to know the exact age, not only because they break through at different times, of course, in a growing young animal, but also you can use that incisor, this first incisor, and you can cut it. I can't do this, you need an expert to do this. You can cut this, and then you can count like tree rings, and you actually can know, even in an animal that is 18, 19 years old, most moose die earlier, but some of them were about that age. You can fairly exactly know what they are, how old they are, and it's very interesting to know the age structure of the population as well. So that was interesting. In moose, we also know how to evaluate by now, after experience over many, many years, how to evaluate the nutritional status of an animal. That's one of the first things we always want to know about wildlife animals. Well, are they under-conditioned, over-conditioned, or did they actually die from emaciation? Yeah. And so in a moose, this is actually the skinned back end of a moose, the gluteal region here, and that is the tail of the base of the tail, and there's a fat layer that you can cut and measure, and then you get a fairly good idea. Now again, in most cases, we want to know whether an animal was starving to death, and we can do this without fancy measurements. I mean, we look for certain types of changes in the fat tissue of the animal in order to determine that. But it's good, again, as a general to have a method of evaluating the nutritional status. Yeah, this is what a collaring of a moose looks like. I mean, we have a spotter plane that flies over the landscape. If they spot a moose from higher up, we can send the helicopter in there. You can see, those of you who do accounting can see how the numbers are adding up suddenly, right? And then they're being actually anesthetized or sedated from the helicopter. And the biologists can do this. There's usually some veterinary oversight because of the drugs that are involved. They are very potent drugs. And then the animals go down. You can get your hands on the animal. You can draw blood. You can, I guess initially they even pull the tooth so they can do the age analysis. Obviously, you want to minimize this time that the animal is on the ground being handled, and so they can do this all in less than 15 minutes. And then nowadays you can reverse the drugs and they can get up and run away. And then you can follow them. And this is a moose. You shouldn't really get so close to a moose. I know they probably used a telelens, but they got closer to that moose than they should have been. And I mean, it's a little bit difficult. It may have a slight head tilt. This was an abnormal moose behaving neurologically. And it was found, either later was found dead, or if it was deemed to be basically moribund, it would get shot by somebody and then it would get examined. And so you can either do a field necropsy. I prefer the winter personally because there are no mosquitoes and ticks. But it's pretty cold at this point. This is about minus 10 degrees Celsius probably, so somewhere like there. Our collaborators put together these field kits, and so it can be done. Of course, if I'm involved, I have to drive up 4 hours and spend time doing the necropsy. Coming back, my director barks at that. Says, no, you don't have time for that. But when I get involved, I like to get involved. So we have a laboratory, and so on some occasions they were able to take out the entire carcasses. One of them they helicoptered out and get them to us. And that is, we have everything there that we need. It's not like somebody forgot the bag and then you have to drive the snowmobile back to the station and get something. And so it was quite interesting. So one of the things that came out of the study is that, yes, quite a few of these animals do have neurologic disease, not only the ones that are reported randomly by the public, but also the ones we followed when we collared these animals. And one thing that you can find in a very, very small number of these animals is a parasite that migrates through the brain, so-called brain worm. And so if you're really tuned in and you pay close attention and you have a good specimen, you can, in 2 out of approximately 140 moose, you could actually find this parasite with your naked eye examination. Now, if one goes and actually examines these tissues, brain tissue, under the microscope, well, suddenly we find evidence of this brain worm in 60 of the 140 moose. Makes a big difference, right? So it's important to look at the tissues under the microscope. And I do love this one. I mean, this looks like just like a happy camper, but this is actually a worm in cross-section. You have the gonads. Worms are basically intestine and gonads. And then you have here the intestine. And so that looks like a smiley face. That is the same worm. It's coiled up in the brain tissue. It migrates through there, damages that brain tissue, and causes ultimately the death of the animal, either by emaciation, because the animal is not foraging properly, or because it falls down somewhere, or it hits a center in the brain that is really important for life function and destroys that. And so now we know that actually the liver fluke, yes, we found a lot of evidence of liver fluke infection in those moose, but we think they didn't really die from the liver fluke problem. They really had a bigger problem with the meningeal worm and, of course, other things. There's no population in the world where you have one thing that is the issue, right? I mean, you have to look at enough animals to really get the full picture. But we know now that this worm is really important in the moose health and disease situation. Now, quite serendipitously, the final host is, again, the white-tailed deer. And there are some different intermediate hosts. Again, they're snails and slugs, but they're different from the one that transmit the liver fluke. And so the issue is still the same. After all we did, the issue is still the same. It's the white-tailed deer population, to some extent, that causes this problem, the expanding range and density of the wild-tailed deer population. And so ultimately, this is just a brief attempt of mine to put it together. Well, we have predators that do play a role, so I'm not going to deny this. There's your Paul Bunyan, right? I told you about him before. And wolves, bears, but then there's the white-tailed deer that carries diseases, and there are other diseases as well. But I do want to point out the worm in the brain and the liver fluke. But there's also climate change. So in this study, they also employed a sensor. They gave it into the rumen of the animal that would record the temperature. And so we know what the comfort range of a moose is. And it turns out that in the summer months, often, sometimes even in the winter months, the comfortable upper limit for a moose was exceeded quite often. And when that happens, that puts a physiological stress on the a metabolic stress on the animal, and it can decrease the time that is actually eating, going out, foraging, because it likes to spend the time in the shade somewhere rather lying down than actually eating. So there is a risk they are underconditioned. And then it also has elevated heart rate, elevated respiratory rate, and all of this has a negative effect on these moose. So that is another outcome from that study. And plus, well, the increased temperatures allow the white-tailed deer to actually exist in an area where they didn't exist before. When you have more white-tailed deer, you have more wolves. If you have more wolves, well, there's also a chance encounter that they see a moose and take it, or a moose calf and take it. Right? So all of this factors into this. Of course, there's not just one reason. There are multiple reasons. And so the question then always is also, well, why are you doing this? What do you do with this information now? Well, you can try to do something about the things you find out, right? Can we do something about climate change? Well, obviously that's a big ask, and something we still have to figure out somehow. But maybe we can reduce the white-tailed deer population, which is the natural host for some of these parasites. So we could increase the hunting season. We could try to do something. We could reduce them with sharpshooters, theoretically, right? And then, can we control the predator population? Now, that's also difficult because the wolves are, as I mentioned, a federally protected animal. And so there is a hunting season in Minnesota, but it's fairly limited. And of course, there is always a very big discussion about the wolf population. I think there's no other animal on the planet where there are so many opposing views. Maybe wallabies are one, maybe I'll come to find out here, but I mean, the wolf is certainly one. And so And again, they figured out where they give birth. And so you could potentially create habitat that would allow and protect this habitat, protect these sites where moose give birth to calves. And so you could potentially increase the survival. And so that's what I'm mentioning here. But again, the why. Why do we do it? Well, there's also some very practical reasons. Our state was free of tuberculosis for about 40, 50 years. And of course, everybody cherishes that situation. Now, in 2005, a cow was detected that actually had bovine tuberculosis. And I know you in the British Isles know exactly what I'm talking about when suddenly bovine tuberculosis shows up, right? And so, unfortunately, this was detected in the neighboring state where there's a healthy competition, but that's just one side effect on it. But I mean, what was done is, shortly after the cow case was identified, well, we found white-tailed deer, free-ranging white-tailed deer that had the same issue. And of course, that is a real problem. Now, suddenly, we have a wild animal population that has a disease that we greatly care about from an economic aspect of the state and of the country. And fairly quickly a study was done where we then looked further into, I mean, how many of these white-tailed deer are carrying it and how can we eradicate this. And I mean, I guess up to now, I've never really touted our horn here, but in 2012, just 7 years later, the state was declared tuberculosis-free. And that hasn't, and that's because we went in and looked exactly where the disease is. We found it early because we had a continuing wildlife disease monitoring program. We found it early, and when we found it, well, the measures were taken, sharpshooters, whatnot, to reduce the population of white-tailed deer. Of course, on the agricultural side, there were measures taken. And so within 7 years, the state was declared tuberculosis-free. Michigan, up to this point, another state on the Canadian border, has been battling with this issue since about 2000. And it seems like they are nowhere near of actually eradicating tuberculosis from their state, because they detected this later, right? And I mean, the reason why we detected it earlier is because we saw what Michigan was struggling with. And so that's what I'm saying here. This is a lung that has lesions, changes that are indicative of tuberculosis. This is a lymph node that has a focus of inflammation here. And when you do a look at this histologically, again, that's what pathologists do. We see this inflammation here with these large cells. And when we do a certain stain, we can find the tuberculosis bacteria in this section. And then we just have to still submit samples to a bacteriologist to tell us that, yes, this is Mycobacterium bovis. Now I'm coming to the wallaby, maybe a little bit later than you were hoping for, but I wanted to set the scene. And I will admit that we haven't looked at a lot of wallabies. We have done a very small pilot study, if you want to, a feasibility study. And so I want to talk to you about that right now. I know you know a lot about wallabies. The wallabies, they come originally from the eastern coast of Australia, and there's a subpopulation on Tasmania. And from what I understand, it's not quite clear whether these wallabies that are here actually come from Tasmania, which is the Bennett wallaby, it's a subspecies, or from the eastern coast of Australia. But the wallabies, you have been very welcoming to us, Anna and myself, when we come, but you apparently have also been very welcoming to the wallabies. And so they have, as you know, at this time established a self-sustained population, not only on the Isle of Man, but also in Scotland, maybe England now, Ireland, northern Germany, as I find out, and also in France. So they're very good at that. They are marsupials, of course. They have a vegetarian diet. They can be up to 80 centimeters tall, which makes it the tallest terrestrial mammal here in the Isle of Man. And the females are a bit smaller. They can live up to 10 years, from what I understand. In my reading, it was said they often live solitary, but they can form mobs in areas that are really suitable to them. They have a gestation period of 40 days, at which point the embryo leaves the uterus and cloaca and climbs up to the pouch. And in that pouch it spends, and these are called joeys, and then they can spend they will spend about 280 days in the pouch. 220 days they're exclusively sitting in this pouch, but after that period they come in and out. And after 280 days, they basically leave the pouch permanently. And so they're fully grown at about 5 years of age, but they have sexual maturity at about 2 years, a little bit less than 2 years of age. And they can live to about 10 years in the wild. At least this is probably data from Australia. I was told, well, if you want to see a wallaby, when I was first here, you probably don't have to go to the wildlife park. You should just go to Close Hartfield and see whether you can find them. And lo and behold, I did find them. And I mean, there's one sitting right looking at me. I was a little bit surprised how they're not very shy, apparently, or this is a neurologically impaired or vision impaired animal. I'm not sure. As we, of course, mentioned already, maybe earlier, I'm not sure, in talking, well, there are opposing views to wallabies. Some of them think, well, they're not a native species. Maybe they shouldn't be here. Others think, well, there may not be a native species, but maybe they attract tourists. And then, of course, there are farmers that also have views on that, because these animals, well, because they're free-ranging, they move from one farm to the other one, and potentially they can spread diseases that way. And so, of course, that's the question that got me involved. And if you want to make any decisions, if anybody wants to make any decisions, it's good to be informed. So that's why also the density study was conducted. And so, yes, there is a thermal imagery map that was produced by BH Wildlife Consultancy, and just recently, at the beginning of last year, maybe in 2023, if I remember correctly, focusing on the main area where these wallabies are. In the meantime, I heard that yes, there are sightings even at the southern part of the island, but that most of them are here in this area around the TT course, I guess, in a way. Not quite sure, somewhere there. And yes, there are, as we know now, approximately 1,000, maybe even more wallabies on the island, free-ranging. And there were also reports of, well, we see a lot of dead wallabies, and we see animals that are neurologic and that have ocular opacities, like the one seen here, taken a picture of by Kerry Jenkins, that kindly enough provided that picture, where we have an eye that is clearly abnormal. And there are various reasons why that could have happened, and there probably are multiple reasons. Could this animal have a trauma? Could it have run into something? Could it have a systemic disease that affects the eyes, or another disease which just focuses on the eyes? That is all possible, and you can only find this out by getting your hands on that animal and examining these animals. So we kind of put our heads together in good Manx fashion, I guess, over a cup of tea, and started talking about whether we can actually find animals and conduct post-mortem examinations. And so questions were, well, do they have an abnormal high rate of death per time period? Is this a healthy population, or is this maybe not a healthy population in general? What is maybe the cause of these neurologic signs and ocular disease that was reported anecdotally? Which infectious diseases do they carry? Which parasites do they carry? And could have these diseases, could they have an impact on farmed animals or pets or humans, right? And then again, it would be interesting to see how inbred is that population. I don't know whether I ever got a number of how many animals originally were imported to this country and then escaped. But we have to assume that there's some sort of a genetic bottleneck here, and that can be further analyzed by conducting genetic studies. So our study, and again I'm talking about the mini pilot study, included 3 animals. We started last year. 1 animal was found dead. Again, I have a little bit of a hard time estimating the age of a wallaby because I'm not really used to the wallaby like I'm used to a wolf or to a moose or something, right? So you can go by size, of course, but as we know, this can vary quite a bit. So I'm not quite sure about the age of these animals. They were was found dead. This was in a fair postmortem condition, I would say, not prime, but okay, we could work with it. And 2 of the 3 were euthanized via gunshot because of neurologic disease that the person noticed. And so they were in a good postmortem preservation. 1 of the wallabies was a male and 2 were females. And so the only thing that we wanted to do was get our hands on it, perform a pathological examination, a necropsy, or whatever you want to call it, post-mortem, and then collect some tissues for histopathology. And the reason was that I'm from Minnesota, right? So I would have to take those tissues with me. And of course, Minnesota and the US are a little bit worried about what I can bring in. And once it's fixed, the good thing about fixing in formalin is that there shouldn't be any live agent in there anymore. And so I can bring fixed tissues to the United States. But if one wanted to get more serious about this and invest more money in it, well, one could also preserve tissues that are not fixed, they're just frozen, and then you can actually look a far more thorough investigation of what agents are actually present in these tissues. And again, I'll show you maybe the limits of what I could do in the next few slides. So these were the animals. These are actually the 3 animals. This is a field necropsy. All of them are field necropsies. And so you may notice that our biosecurity was a little bit suboptimal, but for that kind of purpose, it works. We were not really expecting to meet a highly contagious disease that can affect humans and livestock. And well, we will find out what we found, but this was reasonable thinking. The first thing we saw by performing the necropsy was, well, these animals have worms in their stomachs, right? And so this is the stomach is a little bit different in a marsupial, in a kangaroo or wallaby. It has these succulations that's different from other stomachs. But here through the wall of the stomach, you can already appreciate this worm. And there's another one, and there's probably another one. This is actually a hair, so this is not a worm, but these are the worms. When you cut it open, then you find quite a few of these worms in the stomach. And there are worms in stomach of livestock, in worms of sheep and goats, that can actually be quite pathogenic. I don't know whether you have this problem here, but we have a problem with barber pole worms in the United States and also in Germany. And so you start wondering, is this a worm that only can be carried by wallabies, or is this a worm that could also potentially be spread to livestock, right? And in order to find that out, you need to actually team up with a parasitologist that can tell you exactly what kind of worm it is. I could tell you this is not the barber pole worm. I can tell you this by looking at it. But if you want to know exactly what it is, you have to employ a parasitologist. And fortunately enough, there's Tani Carpenter here from the Exen Parasitology Laboratory in Douglas that can do just that. I don't know, Tani had said that she might be joining us here this morning. I'm not sure whether she did. But thank you to you. We took some of these worms and submitted them to Tani, and she determined that this is a rugopharynx species. And which I hadn't encountered before in my work with wild animal species. And I do occasionally see wallabies and kangaroos in the United States that are being kept there. And so, yes, then you have a name for a parasite, and that's good. With that information, I did contact my colleagues in Australia that I know, and wanted to know whether they can actually spread to small ruminants, for example. And no, they don't. So that's good news for the small ruminant population on the island. They do not spread to small ruminants as far as we can tell. Not in Australia, probably not here. The other thing is interesting, of course. Well, obviously, I think these worms has been basically perpetualized in the Manx population. They came with the first wallabies that were introduced to the island, and they have not left the island. They have, over the 60, 80 years that these wallabies are here now, they have actually also maintained the population among the wallabies. I think that's quite interesting. The other thing that we noticed is that the esophagus lining in two animals looked clearly abnormal. This is what a normal esophagus lining looks like. So esophagus is basically the connection between your oral cavity and the stomach, right? So this is what it should look like: smooth, white. This is what it looks like histologically, a normal esophagus of a kangaroo here. Just a few layers of this lining, and then some muscle cells that move the food bolus to the stomach, right? Now this is what it looks like in two of those kangaroos. It looks like a green velvet, essentially, and so clearly abnormal. When I looked at this under the microscope, well, this wasn't really that smooth, but it actually had all these fronds. The lining was clearly thickened, quite remarkably thickened, had all these fronds, and these fronds were lined by organisms. So these, and when you look closer at this, you'll notice that these are actually fungi, maybe something like a candida, it's hard to say, and bacteria. And so essentially what these animals had, these two animals, was an esophageal thrush. Maybe some people know this condition as a thrush, right? And I mean, in thinking about this, this is probably not their primary disease problem. All these animals, by the way, were under-conditioned in my estimation, because I didn't see a lot of fat in the subcutis or hardly any fat in the body cavities of that animal, of these animals. And some of them turned out to be having a lesion that would almost indicate that they could have died of starvation anytime soon. Now, two were shot, one was found dead. So the one that was found dead potentially died from starvation. Although I'm being told that the vegetation is plentiful here, and they did have, of course, contents in the stomach that indicated that they were eating. Yeah, and so then, of course, remember, these animals, two of them were reportedly behaving abnormally. So the brain was of great interest to me and to the wildlife biologists. And so this is a brain of a wallaby. I don't expect you to know what the brain of a human or other animals look like, but it has a fairly peculiar shape, quite interesting. And so I basically took a cross-section here from the forebrain, and also cross-section here through the cerebellum, so the hindbrain and the brainstem. And so that's what we see here in this picture, a histological depiction. This is the brainstem, this is the hindbrain, cerebellum, and this is from the forebrain, a certain area of the forebrain. And the keen pathologist noticed already here at this magnification that there is some blue here that is more than it should be. And here in this area potentially, when you look closer, you'll find that the brain, and this is from the third animal that we examined, it had the best lesions for a pathologist, and it was the freshest. And so I'm showing you this animal. It has these inflammatory cells that are in the brain, so this is an encephalitis. If you find them in the meninges, it's a meningoencephalitis. And then you start wondering, well, why does it have the inflammation, right? What are they doing there? And this type of inflammation suggests most likely a viral infection or an infection with a single-celled parasite. And so you look around and you look for that single-celled parasite. The viruses you cannot detect. There are some clues that you can potentially find, but you cannot find the virus. It's too small, even for the light microscopy. But you can find this single-celled parasite. And as it turns out, by scanning the slide a little bit more, yes, there were single-celled parasites in this brain tissue. And then you see this, and at this point, you wonder, could this be Sarcocystis neurona? So there are various parasites that look like that. Sarcocystis neurona, Neospora caninum, which can affect cattle and sheep and goats. Sarcocystis and Toxoplasma gondii is one of them. So this would be the three big ones, right? And then you have to employ different methods to find out which one it is. And then remember, this animal had also impaired vision, or presumably an impaired vision, and the reports were there of animals that had impaired visions. And so I looked at the eye as well from these animals, and this is again animal 3, and we can notice that there is an inflammation in the iris, but also in the retina. And when you look closely at the retina, suddenly you find the same parasite also in the retina. And so maybe there's an explanation, at least for some of the cases, that it is caused by a parasitic infection, the clinical signs and the lesions. And in this animal 3, I mean, I just looked at the heart muscle, there is the parasite in the heart muscle. I looked at the skeletal muscle, there is the parasite in the skeletal muscle, even again causing some degree of inflammation. And so you always want to remember what I look at histologically is really just a very, very small part of this animal, right? So if you see them, well, that is indication there's something going on. And the nicest one, and that one really makes my heart jolt a little bit, is the one in a big neuron near the adrenal gland. And so they do tend to target neurons, muscle cells, but also other cells, this parasite. And so just to summarize it, we employed different methods. So all wallabies were underweight. 2 of the wallabies had these stomach worms that were identified as Rugopharynx species. And so all 3, as it turns out, had some degree of encephalitis. The third one that I showed you, obviously most picturesque one, had the heaviest encephalitis, but the other 2 also had encephalitis. And again, then you start thinking, wow, this is quite something. If you only look at 3 animals and all 3 have encephalitis, which is ordinarily a pretty rare condition, that's odd, right? And so this one also I showed you the eye lesion, no? The endophthalmitis. And then we employed some additional testing that can be successful in formalin-fixed tissue, but doesn't have to be successful. It's better to use non-fixed tissue for this testing, but we still did it. And it turns out that we employed this test for Toxoplasma gondii, a molecular test, and it was positive, not only for the 3rd animal where I actually saw the parasite, but also for the 2 animals where I never saw the parasite under the microscope. And then there's another test that you can use to corroborate your finding. In the best of all possible worlds, all tests are going to be positive, right? But in reality, often there are some discrepancies. And so with this test that shows the parasite stains it, if you want to, in the tissue section, yes, we saw it in the 3rd animal, but I had already seen the parasite. Now I know it's this parasite though. And I didn't see it in the other sections. So there's still a question mark here whether Toxoplasma gondii is actually the culprit or whether it was just there, you know. And so these were my main takeaways, our main takeaways. Well, encephalitis is apparently common, but we only look at 3 animals of 1,000 or 1,200, right? And maybe a population that was pre-screened because they were neurologic. And so in one animal, I feel very confident that it was actually caused by this parasite, Toxoplasma gondii, while in the other ones, I have some question marks. And just because it has Toxoplasma gondii doesn't mean it couldn't have something else that either amplifies the effect of Toxoplasma gondii or is even independent of Toxoplasma gondii, right? And so these other things, and we can talk about what they could be in the next slides, this would be worth examining. Then we have the gastric endoparasites, I mentioned that already, and the esophageal thrush, which I think is secondary to this animal being sick, probably secondary to not eating, wanting to eat, and didn't really cause the issue, right? Now, if you think about Toxoplasma gondii, it causes a disease condition that is called toxoplasmosis. And we actually know that Australian marsupials are highly, highly susceptible for this disease. I mean, I guess the assumption would be, yes, they evolved in Australia where they didn't have any, well, that's the thing we haven't talked about, any cats, because the cat is a definitive host of Toxoplasma gondii. So they evolved in an area where there were no cats, so they never were exposed to this parasite over the, whatever it is, millions of years, right? And now we take them out of Australia and put them somewhere where cats are, and they can develop this disease, and they're highly susceptible. There are other species that are highly susceptible: New World monkeys, lemurs, Pallas cats. And interestingly enough, also, as it turns out, sea otters of the Pacific coast of North America. And why that's interesting, we can touch on a little bit after this slide. Now, the parasite is a single-celled parasite, and a definitive host is the cat. And it doesn't matter whether it has a tail or not. The definitive host is the cat, including the Manx cat. And in the cat, it lives in the intestine and amplifies in the intestine, but it doesn't cause any harm to the cat. You will never know that the cat has Toxoplasma in the intestine unless you do testing of the fecal matter. It doesn't cause any harm. Now, it does shed infectious stages into the environment that is then being picked up by intermediate hosts. And the thing about Toxoplasma gondii is that it's not very selective. It actually has a really broad range of intermediate hosts that it can use. And obviously, from a parasite standpoint, that's wonderful, right? You don't have to rely only on one species. Well, you can spread it out, even between birds and mammals. And unfortunately, humans can also be intermediate hosts. And in those intermediate hosts, the parasite then does a further development in its life cycle, and it will ultimately form these cysts that I showed you in the skeletal muscle, right, that a predator would eat later on, or in neurons that are all throughout the body, and in the brain particularly, of course, and spinal cord, but also all throughout the body. And so a predator that is now like a cat that is eating on this will ingest the intermediate stages, and the parasite can complete its life cycle in the cat. Unfortunately, under certain circumstances, the parasite in some of these intermediate hosts can cause some individuals, some species particularly, can cause disease, again, toxoplasmosis. And I mean, the best known complication in humans maybe is miscarriage in women. Yeah. And it doesn't happen that often, but it can happen. This is a single-celled parasite. The final host is a domesticate. Humans, the issue is miscarriage. In some humans, even depending on what kind of strains of Toxoplasma gondii are involved. I'm being told in South America, for example, we have strains that are very, very pathogenic, very virulent, that can cause particular severe disease. And so miscarriage due to Toxoplasma gondii is quite common in South America, as I found out, and also systemic disease, like our kangaroos have, can happen in humans in South America. But it's not really that common here in Europe and Asia. The parasite is everywhere where cats live. So usually we have worried about immunologically naive women that are immunocompromised. The common recommendation usually is you should not get your first cat when you're pregnant the first time, right? Don't get the first cat in your life when you're pregnant, is a take-home message. Because when you clean those litter boxes, well, you're going to be exposed to feces, and even if you're very careful, you might actually ingesting this parasite somehow, right? And then there's another way of getting it, which is probably very rare, is eat undercooked meats from intermediate hosts that are infected. Sheep, kangaroos, wallabies, whatever it could be, right? And again, in South America particularly, we are worried about, or they are worried about, insufficiently cleaned vegetables. Cats have maybe gone into the vegetable garden, have done what they do, defecated there, and then the rain and all of this resolves, spreads it around, and now it's on your vegetables, or maybe even in your water. And so you could potentially, somebody could potentially become infected that way. But it always comes back to the cat. It's where the cat is. It's not necessarily where the intermediate host is, it's where the cat is, where the, right? And so this here particular, again, the water source, there I'm circling back to the sea otters of the coast of North America. Well, they get obviously infected because there's so much Toxoplasma gondii in the coastal landscape that runoff water from the rainfalls flushes these into the ocean, where the sea otters live close to the coastline and they become infected. Well, toxoplasmosis is nothing new to the Isle of Man, as it turns out, when you look a little bit around. Well, there was this is a paper, a retrospective study of cases in zoos, including the Wild Animal Park here on the Isle of Man, conducted by a group in Leeds. Mark is a wonderful pathologist there, and Daniela as well, but I don't think she's there anymore. And so they diagnosed systemic toxoplasmosis, something similar to what I showed you in this last kangaroo, wallaby, that affected small-clawed, Asian small-clawed otters in the Wild Animal Park here on the island. And so they described the lesions, but I mean, one thing that they basically pointed out, it is highly likely that the outbreak relates to a high oocyst burden. That's the infectious stage that the cat actually defecates, right? That is maintained by the felids and a range of susceptible intermediate host species in this area of the zoo, or of the Wild Animal Park. Right, so it's nothing new really. I guess, I mean, conclusions may be the wrong word, but I mean, if we look forward, if we really wanted to understand what's going on with the population, I think the first three animals gave us a glimpse, and it tells us, well, there is a disease issue with this population. It may not be a healthy, thriving population, although they are increasing. There are health issues with this population. But we definitely need to look at more animals. If you look at more animals, you will find different kind of diseases. Again, I told you there cannot be a population that has only one issue, right? So, I mean, how many animals you want to look at? Well, if you have 1,000 animals, depends on what the prevalence of that disease in that population is. If it's 10%, well, you might have to look at about 100 animals to discover it. If it's 100%, well, you only need to look at one animal and you know what's going on. But again, that's not going to be possible. And again, I mentioned we could employ other testing modalities. Those two animals that had encephalitis, but didn't have any cysts, any parasite in it, could it be that they have an underlying viral infection? We know about viral infections in wallabies. There could be an orbivirus that they have in Australia. There could be Rustrella virus that was recently discovered that affects wallabies. There could be looping ill virus potentially. I don't know whether it can affect wallabies, but one could look. One could do serology, and one could employ the molecular techniques to find out whether this virus is there. And there's tick-borne encephalitis, something that is potentially coming over to the Isle of Man or has already been here. I don't know exactly, that we could look for with these tests. And then, I mean, if one had health data on livestock and on humans and pets, well, one could compare what one finds in the wallabies and in the other populations on the island and see whether there are any overlapping issues, right? Again, I mentioned genetic testing already multiple times, probably very interesting to conduct this. It's not something I could do, but there are geneticists out there, biologists out there that can do this very easily, and they can, it's amazing the amount of information that you can draw from this testing. And again, one could potentially look and catch living wallabies and get samples from them and assess their health this way. But again, there's always a risk, of course, that somebody gets injured. In the case of the moose, of course, there's also a risk to the people who capture them. They could get injured. In a wallaby, I'd like to think probably the risk is kind of minimal. Yeah. This is just throwing this out there. I mean, there is a threatened population of brush wallabies in Australia, and they can be equipped with monitors that emit signals. So it could be done, but that's for other people to decide on how important that is. And with this, I guess we employed ChatGPT one more time. And so the tail of the wallabies and the cat without a tail. Is it really just something, toxoplasma from the cat, that is basically affecting the wallabies? I can already tell you, probably not. There are other things. Again, you just need to find them. And this is probably not the end. And with this, I gave some additional image credits for pictures I used that I didn't really properly quote, but I do want to do these people justice that took these pictures. And again, I'm very thankful for being allowed to be here and to be getting involved with this study. And I probably took up too much of your time, but I hope it was entertaining and helpful to you. Thank you. If you have any questions, please feel free to fire away. Again, I'm a pathologist, so I look at a fairly narrow picture here of the whole issue, but I'm happy to answer everything that I can.
Ask one question, please. The red-necked wallaby, or another name for it is Manx wallaby, was believed to have originated from Tasmania. Now, I'm only thinking, when you look at what the wallabies are doing in the Isle of Man, if you go to where they originated from in Tasmania, the population there, I think, would be in millions. Can you learn anything from speaking to the Forest Service in Tasmania about what problems we have there and how they get on with them? Would that be helpful?
Right. Again, I tried to reach out to Australian colleagues to find out about that gastric worm. And yes, obviously, that would be helpful. Now, I think, again, just on a different note, the Bennett wallaby is a subspecies of the red-necked wallaby. From what I understand, I'm not quite sure whether the Manx Wallabies came from the Bennett's wallabies from Tasmania, or whether they came from the eastern coast of Australia. But the point is well taken. Yes, I mean, of course, it will be good to interact with the Australians because they know a lot about wallabies. And again, I try to do this just informally in a way. And again, if we talk about the mortality rate, well, what do we compare it to? Is it abnormally high? Well, we have to compare it to a population, well, somewhere in Europe, but ideally also to the Australian population. And yes, there are a lot of information that one could get by doing that.
You were talking about populations that are a long way away. Presumably there would be some interest in looking at the wallabies that are actually in the wildlife park in Ballaugh, because that's a captive group of animals quite close by.
Yes, I mean, that is obviously true. I mean, I come to understand that there is not really a captive population of wallabies at the Wild Animal Park right now. They're coming in and out. They're basically wild wallabies that are now coming back to the zoo on occasion, but then they leave again. And I mean, I'm assuming that over the years back when maybe postmortems were conducted by somebody, tissues were maybe sent out to Mark. I mean, he's been doing this since 15 years, so I don't know what was 30, 40, 50 years ago. Of course, over the time, there are probably some data. I just don't know exactly how relevant they are. It would be nice to find them, yes.
Thank you. Has anyone looked to see whether any of these parasites are in native species apart from wallabies? Has it spread beyond?
Right, right. And again, I don't think the data are there. I mean, again, this gastric worm, the one in the stomach, theoretically shouldn't be in any native species because they're specialists for wallabies. But of course, you never know exactly until you start looking. But that's the evidence there is. The Toxoplasma gondii, I can guarantee you that the wallaby is not the only intermediate species there is on the island. There are plenty of them. I mean, theoretically, they really are more meant for rodents, right? Because that's what the cats catch mostly. I don't know that there are data out there, but they're going to be in rodents, and again, maybe even in some birds. If you look for it, you will start finding it.
The vets on the island don't seem to be very conversant with what can transfer from wallabies to, for example, horses. I have 3 horses, all of which at the moment have a problem with the esophagus. I'm just wondering if there's anything like the thrush in that could be transferred.
Again, I would assume, I mean, ideally one would exactly diagnose the issue in the horse, of course. My gut feeling is that probably what the wallaby has is unlikely to be carried to horses, but again, that's just a wild guess at this point. Yes. In the end, you need to look and diagnose it, and then one could talk more knowledgeable about it. Yeah.
Well, is it, would it be a fair summary to take away that the wallaby population does not, at least on the 3 animals, seem very healthy, but in terms of risks to livestock or humans, the risk is not from wallabies, but it's from the cat populations out there, whether domestic or feral cats, which are quite widespread? Right.
I mean, again, it's a mini study. Ideally, one wants to expand it. But from what we found so far, it's more what we found is more of a welfare issue to the wallabies than an issue for the livestock and for the human population. What we find when we look at 10 animals, 20, 30, 50, 100, that is a question. Thank you.